All right, good afternoon, everyone. We are going to talk about water, salt, and conservation and regulation today, looking at some of the organs and organ systems that are involved with maintaining osmolarity and water concentration, which we just got done talking about how important that is, right? That cells have the right water and solute concentration, that extracellular fluids have the right water and solute concentrations. We also talked about ways that animals have taken advantage of this, or how organ systems take advantage of this, right? When we talked about cystic fibrosis, for example, and how we transport water in the epithelium of the trachea from the interstitial fluid to the mucus layer to liquefy mucus. Here's another great example of that. Blue crabs loosen up and split their old exoskeleton by concentrating solutes in their body. So there are certain hormones, cortisol in particular, stress hormones that are involved with this. They release a whole bunch of that when their shell becomes too small for their growing bodies. This will cause them to bring in a bunch of salt or pump salt into their body tissues. Their bodies swell, right? And they are a little bigger than they normally would be. This puts tremendous stress on the old exoskeleton and cracks it. And they climb out of that exoskeleton, right? Old exoskeleton with a new, softer one. They're swollen, right? And as they harden, they then shrink back down to normal size, and that leaves a little space inside of the new shell, which allows them to grow again. And so again, by taking advantage of osmolarity and osmosis, the movement of water, they can stretch and break their shell, and they grow into their new one. So this is mostly in chapter 27, I think, in the new text, the textbook I've given you. And this is our first example, the example they use kind of our integration exercise here. And you can see the splitting of the shell here after the blue crab has swollen from taking up solutes and allowing water to swell its body. So these are kind of the topics we're going to cover here. We're going to talk about osmoregulation and whether you're a regulator or conformer. Vertebrates like us are conformers. A lot of invertebrates are regulators as far as their water and solute concentration compared to the environment. Talk about body fluids real quickly, make sure we're on the same page. And then we'll look at some patterns in animals that we see. And then at the end, we'll talk about the kidney, right? We'll talk about the kidney and the importance of the urinary tract in the regulation of osmosis. So remember, water is huge. Well, without water, there is no life. Water is very unique in its ability to form partial positive and negative poles on the oxygen and the hydrogens, right? Which allows them to have all of the special properties of water like high specific heat, the ability to dissolve solutes that are polar, right? And to carry those solutes around. Then water concentration becomes important with animals because water also is involved not only in hydrating our tissues and our body fluids, but it also participates directly in chemical reactions by building and breaking a lot of the large bonds between our larger molecules that we call macromolecules. So water is very, very important. A matter of fact, and as far as the evolution of osmoregulation goes, there's a whole bunch of organs that work together to maintain the right water concentration in our bodies. And that just right there, and the amount of energy, the maintenance that goes into those organ systems is very high. So maintaining proper osmolarity is very important in animals that occupy very different niches. As a matter of fact, temperature and osmolarity are two of the major environmental extremes that animals have hard times moving from one extreme to the another or one environment to the other. And there are many animals that can do this. And if you can move into a environment that has a completely different osmoregulatory pattern or requires different osmoregulatory organs, and you can move into that environment and feed and be successful in that environment evolutionarily, then you're taking advantage of a niche that other organisms can't. So evolution has driven a lot of our ability to be osmoregulators. A wonderful example right here with the marine iguana, one of the few lizards that actually lives and spends most of its time in the ocean. And so there are very few lizards that can do this, and it feeds in the ocean. And this one's basking, and they can do this because they have salt glands that are associated with the nasal cavity. They can drink salt water, and they can remove the salts from that water, and they sneeze them out. That's why the heads of marine iguanas look white. They sneeze the salts which collect on their heads. So we'll talk about several of these types of adaptations. Also, think about the move from water to land, right? To become tetrapods. Both insects did this. They did it first, right? A lot of the arthropods did this first, and then vertebrates moving from water to land. Land is very hypertonic. All right, it has a lot less water in it than our bodies do for the most part, and so we had to come up. Our evolution had to evolve some mechanisms that allow us to prevent water loss through our skins and our kidneys and some other structures that we will talk about. So osmoregulatory adaptations had to occur in order for organisms to move on to land. Insects did it first. They did it very successfully. They have very thick exoskeletons. They can close off their respiratory tubes to the environment so that they don't lose excess water. And they can also dehydrate their feces in their urine. And so they're very good at that. Without insects doing that, a lot of the plants that you see today would never have evolved because plants and insects have that relationship of pollination. The flowers, particularly flowering plants, produce nectar, and then they produce sperm that we call pollen, right? The insects collect that pollen, they eat it, they collect the nectar, they eat it. They also transfer nectar from flower to flower and allow for cross-pollination. So that symbiotic relationship of pollination between insects and plants are very important. Without the insects evolving to become terrestrial, those plants probably would never have arose evolutionarily. So lots of success stories here when we'll talk about kind of how organisms and how evolution came up with mechanisms to control osmoregulation. So as we know, there are several different body cavities, right? So we have cells. Cells are separated from the other fluids or the other environments around them through the cell membrane. So the intracellular environment is the largest compartment in our bodies. If you think about it, we are billions or hundreds of billions of cells. And so those cells all have a volume of fluid in them called cytosol or cytoplasm. So most of our weight is intracellular fluid. And then the second major body fluid is interstitial or intercellular fluid, what we call our internal environment. And then we have some blood plasma. And we could throw another little graph up here for lymph, right, which was found in lymphatic vessels. But these are the three major fluid types that you find in the body. And these fluids are about 60% water, anywhere from 60 to 70% water, and then 40% protein, lipid, and other solutes that are dissolved in it, right? Our internal environment is our interstitial fluid, and that's where homeostasis is maintained. And one of the major homeostatic controls are osmolarity, right? Maintaining our 300 milliosmoles, which we've talked about. Maintaining that body fluid as a tetrapod, a mammal. The other thing I want you to realize is that the extracellular fluids at least give rise to each other. So this is just a picture of the smallest blood vessels in circulation. The smallest blood vessels in circulation are called capillaries. So here are some capillaries, right? It's a capillary bed that's out in tissue. Not only are capillaries the smallest of the blood vessels, they're only place where exchange happens between the blood and the body cells in a vertebrate like us with a closed circulatory system. So this is where we exchange oxygen for carbon dioxide, sugars for waste products. It's where we also exchange water for solutes in some cases. If we're talking about the kidneys, for example. So the cells here, each of the tissues cells has intracellular fluid, and then the extracellular fluid around the cells is called interstitial fluid. Plasma often leaks from these blood vessels. The plasma is the water of the blood, and replaces or replenishes the interstitial fluid. These capillaries can also absorb excess interstitial fluid. All right, so the interstitial fluid is actually circulated just like plasma is. So these fluids don't sit still, and this leads to a term we'll use when we get to circulation called bulk flow, where the plasma is leaky. It actually leaks into the interstitial space and is also then reabsorbed back into the capillaries so that the interstitial fluid is continually recycled also and replenished so that glucose, oxygen, and the supplies that the cells are needed are continually being delivered to those cells. And then the waste products and carbon dioxide are continuing to be swept away and enter back into the bloodstream and then circulate away so that these cells are in the proper environment, getting the proper nutrients and supplies that they need. These capillaries that leak plasma into the interstitial space are called fenestrated capillaries. Fenestrations are the holes or pores that allow plasma to leak. They're very small. They're like a sieve or a filter. Only certain things that are dissolved can actually enter into the interstitial space. This is a picture of a capillary under an electron microscope, showing you the red blood cells in single file. And you can see how thin the wall is of the capillaries. This little circle right here, this little rectangle right here shows you very, very thin pores that are found in the wall of that capillary that are delivering, those are fenestrations, that are delivering the plasma to the interstitial space. This is called capillary exchange that happens between the capillaries and the interstitial fluid. So it's in this interstitial fluid and also in the blood where we're going to regulate and maintain proper osmolarity, right? So to give you some idea, here is a table that you don't have to memorize. It's just a table that shows you kind of a whole bunch of animals, right? And their osmolarities of their extracellular fluid. So they got ionic concentrations up here in millimoles, and then their osmolarity of the environment in particular. So you got habitat over here, and then overall osmolarity of the body fluids. And then we have ion concentrations here. And I've kind of labeled this figure. It's a very old figure from probably the original edition of your book that I took a long time ago that shows you a bunch of animals. So osmoconformers are organisms that mimic salt water. They conform to the solute concentration of salt water. Salt water is over 1,000 milliosmoles. And so you can see here that in black, this first large group are almost exclusively, right, invertebrates, and their osmolarity is 1,000 all the way down to here. Notice the cyclostomes, the hagfish, are also 1,000. And hagfish are the first of our chordates, which include the vertebrates. Hagfish are not considered, and I'll just point these guys out. Hagfish are not considered vertebrates because of their characteristics of being an osmoconformer. All right, so they are very invertebrate-like in their concentration of solutes. Their solute concentration is near 1,000. What I want you to notice is then the rest of the vertebrates are mostly in much smaller concentrations of solute than the habitat around them, right? So I notice that a lot of the tetrapods and a lot of the fish have osmolarities that are somewhere between 200 and 300 milliosmoles. Now, sharks are different. Sharks are osmoconformers also, notice right here. They conform to what salt water looks like at about 1,000. They can also drop their osmolarity to be around 200 when they enter freshwater. So sharks can control their osmolarity a little bit, increase it and decrease the number of solutes in their bloodstream and interstitial fluid, depending on whether they're in saltwater or freshwater. Notice that most saltwater and freshwater fish maintain osmolarities between 200 and 300. They are regulators compared to the environment that they live in. So you got osmoregulators down here. Sharks use urea to increase their solute concentration. They don't just increase whatever solute they want. They increase an organic nitrogenous waste product, urea, into their interstitial and blood fluids and bring that way up in concentration, which is much less dangerous than increasing sodium and chloride, for example. And that's what they are going to concentrate, as does one particular frog. There is an amphibian that can do this. Notice down here, this ranid here, which is called the crab-eating frog. It can increase its osmolarity of its blood up to 830 milliosmoles, which is close to 1,000, and then drop it back down under 300. And they, again, also concentrate urea. The crab-eating frog is the only frog that feeds in saltwater. Think about that. It's the only frog that can live in saltwater, takes advantage of a niche no other amphibian does. It's a beautiful evolutionary story, if you will, or characteristic of this frog that allows it to take advantage of a niche other amphibians can't. So that kind of gives you some idea of the typical osmolarities of some of the body fluids of animals. So most vertebrates are osmoregulators. Most invertebrates are osmoconformers. And then there's the sharks that are osmoconformers and a frog species that is also an osmoconformer in certain instances when it enters saltwater. So we'll talk about those examples, if you will, and then those exceptions as we go through our discussion. So intra and extracellular fluids, the other thing we already know is that even though the solute concentration, say in our regulators, is around 300 milliosmoles, and so is our intracellular fluid is around 300 milliosmoles. The ions that are found there are different, right? So, and we already know this, in most vertebrates that are regulators, sodium is going to be low on the inside and high on the outside. Potassium's opposite of that. It's high on the inside and low on the outside. Calcium is going to be high, and chloride is going to be high on the outside and low on the inside. And then amino acids, that's what A stands for, our proteins that are negative are going to be high on the inside. These differences in their electronegativity, or I should say their electrochemical gradients, right, are going to allow us to take advantage of gradients to do work, right? Like we talked about with symporters and antiporters and the production of electricity. So remember, the cell membrane is our edge or our barrier to our cells, and it's a selective barrier that controls what can come in and out of the cells. Even though the intracellular and extracellular fluids are typically equal to each other in their osmolarity, they're isotonic, right, to prevent movement of water in and out of cells for the most part. The ions that are in each of those solutions can be quite different from one another. There are a whole bunch of organs that are involved in osmoregulation, and they include gills, salt glands, the gut, the lungs, the nasal cavity, the epithelia that line those cavities are heavily involved, or that line the gills or cover the gills or line the lungs are heavily involved in osmoregulation. And those are the structures we're going to talk about. We're going to work our way down to the epithelium in those organs and talk about how they are involved with regulating solute concentration in these organisms. All right, we kind of already mentioned this, but I got a slide on it. So invertebrates are typically conformers as far as osmoregulation goes. They are going to be very similar to the environments they live in as far as solute concentration. If the environment changes, they might fluctuate slightly in their osmolarity, and they can fluctuate to a certain extent by dropping or increasing organic amino acids often. So for example, if the environment becomes more hypoosmotic all of a sudden, the salt water becomes a little fresher, if you will, or has a little less salt in it, a lot of the invertebrates will conform by dropping their organic amino acids in the extracellular fluid. They simply remove it, the organics out of their extracellular fluid, and pull them into their cells. If it becomes hyperosmotic, becomes a little more salty all of a sudden, then invertebrates can increase extracellular amino acids. And that's kind of what they can regulate if they have to kind of conform to or mimic salt water. Vertebrates, on the other hand, typically are regulators, except for sharks, right? And maybe a couple of other vertebrates. And they typically are going to only allow small changes in their intracellular and extracellular fluids to occur no matter what's going on outside of their body. Many of them are going to add or remove potassium intracellularly or extracellularly when they have to. They can deal with potassium much easier than any of the others. They can remove it or add more of it to the intracellular fluid. The central nervous system does not do that. Again, the central nervous system, we don't want to mess with potassium or sodium levels because those levels are involved with the actual production of electricity, which is essential to nerves, to proper nerve function and muscle function. And so the central nervous system acts a lot like an invertebrate does, and it'll increase or decrease their amino acids or their negative protein groups to change a little bit or to conform a little bit, if you will, if there are very small fluctuations in osmolarity. But for the most part, regulators are strict regulators. They're going to maintain the osmotic conditions so that they are similar in the extracellular and intracellular environments, no matter what's going on out in the environment itself. So let's look at some of our differences between our freshwater regulators and our saltwater regulators, and then our tetrapods, our animals that live in the terrestrial environment. So freshwater animals, their body fluids are going to be hyperosmotic to the environment. All right, so their body fluids are going to be hyperosmotic. Most freshwater organisms that are regulators tend to have blood osmolarities that are a little bit below 300. All right, so they're around 300, but they're actually below. They're actually closer to what freshwater is. Freshwater is around 50 milliosmoles. So it's got a lot of water and very little solute in it. Whereas most freshwater organisms are somewhere between 300 and 240 milliosmoles. So they have a lot more solute and less water. So these guys are going to have to deal with gaining water, right? Water is going to want to move in to these organisms, right? Because there's a lot more water in the environment, right? So they're going to have to worry about H2O gain, and then they're going to have to worry about salt loss, right? The loss of salts, because their solute is going to want to move out, isn't it? Because if it's high in the body, it's going to want to diffuse out. And they're going to lose some salts over their gills and out with their urine, right? They're not going to be able to absorb all the salts that enter the urine. So they are going to actually have to do some things to their body that prevents water gain and salt loss. So their integument, their skin, is typically impermeable, right? So it's going to be impermeable to most salts and to water itself. The gills are actually going to pump salts in to the body. All right, so they're going to pump salts in because ultimately they're going to lose salts from their urine. All right, and so I don't really need that arrow there. So they're going to lose salts through their urine mostly because the urine, the kidneys filter blood, right? They allow blood to leak or plasma to leak into the nephrons, into the kidney tubules, and then you're going to get rid of urine. They're ultimately going to, even if their kidneys are pretty efficient, which they're really not that efficient, they're going to lose some salts. And most of these guys, since they are going to take up water through their gills, are going to produce a very dilute urine, and they're going to basically be urinating 24 hours a day, getting rid of all the water that's coming in. Because of that, they lose salts with that urine. And their gills are now involved in not only moving oxygen and carbon dioxide, but are also involved in pumping salts back into the body so that they can regain the salts that they lose. So their kidneys are typically going to try to conserve sodium chloride, and they do that pretty well, and they're going to produce dilute urine, right? And so they're going to lose that water that they're taking. Most of these freshwater fishes and amphibians don't have to drink because they gain all the water they need over their gills. So the gills of fish, the skin of amphibians, the bladder of turtles, freshwater turtles, and the kidneys of mammals all have the ability to pump salts back into their bodies, right? So they don't lose salt to the urine. So then these amphibians, it's the skins that are really good at pumping salts. In fish, it's the gills that are really good at pumping salts back into the body because they're losing salts. So that's kind of how freshwater organisms work. Saltwater organisms are going to deal with the opposite of that, right? They are going to be in an environment that is going to be hyperosmotic to their body fluids, right? So their body fluids are going to be around 300 or above 300 typically, and the environment around them is going to be 1,000 milliosmoles. And so these guys are going to have to worry about now losing water, right? Losing water. So water is going to come out of their bodies, and they're going to have to worry about gaining salts, right? Or salts coming into their bodies. And so they are going to have that major problem. And so fish, for example, are going to, again, have skin that's pretty impermeable to water loss and to salt gain. They are still going to have to worry about water loss, right? Water is going to be lost over their gills, believe it or not. And some water is going to be lost through their kidneys because they have to produce urine. So a lot of these animals are going to have to drink salt water. And when they do drink the salt water, what they're going to have to do is get rid of the salts. So there are several organs, new ones and modified ones, that their job is to remove the salt that they actually consume when they're drinking water. And so again, these animals are going to try to produce a very dilute urine with very little water in it, so they lose salt through their urine. They are going to also pump salts out, try to pump the salt out. And then they're going to have to drink water because they're going to lose water over their gills and with their urine. Now, as you know, there are some chordates like the hagfish, which are osmoconformers, right? And so what that means is that they are going to have to regulate their body fluids so that they are the same, right, as saltwater themselves. So these osmoconformers, like hagfish, what they do is they are going to increase sodium and chloride. So here's a hagfish down here. It's a picture of a hagfish. And what hagfish do is they increase the amount of sodium chloride in their body tissues in order to maintain the same osmolarity as salt water. They do regulate some of the ions and keep them lower than salt water, particularly calcium, magnesium, and sulfates. And calcium, magnesium, and sulfate can really affect the nervous system and how muscles and nerves work. So we want to keep those concentrations way below salt water in the normal range so that muscles and nerves work correctly. So for hagfish, as I mentioned, what hagfish do is they do concentrate sodium and chloride in their body tissues so that their body tissues equal close to 1,000 milliosmoles. And so that's how they can form. Elasmobranchs are going to increase their urea concentration, which is a nitrogenous waste product of protein breakdown. And they increase their urea so that their concentrations of their body fluids, again, equal salt water. They have a rectal gland that pumps out sodium chloride. So it pumps the sodium chloride out of the body. So as sharks drink salt water in order to try to maintain their water concentration, they will remove the salt from that water and pump it out through the rectal gland, which you will see when you open up your sharks. It's basically located close to where the cloaca, where the intestines empty into the cloaca internally. So that's a specialized gland that you find in all of your chondrichthyans, which are your sharks and rays, that pump the salts out of the body, blood, out of the blood where salt had come in with the water that they drink. So when sharks enter freshwater, they'll drop their urea concentration so that they bring their osmolarity down to 200 or below 300 milliosmoles so that they can be closer to conforming to the freshwater's 50 milliosmoles. The crab-eating frog from Indonesia also is going to utilize urea. It can increase and decrease urea, and it can enter tidal pools. It lives in Indonesia on islands that have very little fresh water on them. And so they come out of hiding at night and then enter tidal pools and eat small crabs and other invertebrates. They also lay their eggs, and their tadpoles are really good at maintaining their urea concentrations so that they can be osmoconformers. And it allows them to take advantage of the saltwater environment, which no other frog can do. And so they're very unique in that way. And they kind of do the same thing sharks do. So that's how the conformers work. But remember, there are very few conformers outside of the invertebrates. Some of the primitive chordates, like Amphioxus and Urochordates, and then hagfish. All the other vertebrates are regulators, except for these two examples of conformers here for us, at least. The elasmobranchs, which are sharks and rays, and then this crab-eating frog. Okay, marine regulators. So these marine regulators, as I mentioned, they're going to have the problem of losing water, right? H2O, and gaining salts, right? Salt coming into their body. And they're going to regulate their body tissue so somewhere between 300 and 380 milliosmoles, where salt water is 1,000 milliosmoles. So like most vertebrates, they're going to be hypotonic to seawater, and they tend to lose water, especially over their gills. They might lose a little water when they urinate also. Because they're losing water with their urine and over their gills, they have to drink salt water. So they're going to consume salt water, and then they're going to have specialized structures that have the ability, or modified structures that have the ability to pump salts out of the body. And that's going to happen in the gills. The kidneys are going to produce concentrated urine when they can, all right? And so they're going to put a lot of salt into their urine and try to remove the water. They also will pump salt out through the gills. Gills have specialized cells that are called chloride cells, and these chloride cells have the ability to remove salt water from the bloodstream and pump it into the environment. And in this way, they can drink salt water because they're going to lose salt over their kidneys and lose salt or pump salts back into the environment over their gills. Water is going to escape over their gills as they're breathing in oxygen for carbon dioxide. And we will talk about these these specializations to the gills and specializations to the kidneys as we start giving examples of kind of how these organs work. The integument, the skin, again, is going to be impermeable to water and to salt gain or loss. And you can see the physiological changes to the environment in some of these animals, because some of these fish can move from salt to freshwater, which is pretty amazing during certain parts of their life history. And eels are a great example of this. Some of the specific salmon and the eels, they can enter and leave fresh and salt water. And sometimes they change color when that happens. So eels, for example, when they enter salt water, their integument becomes impermeable to water and it changes color at the same time because of adaptations to the skin or changes in the skin. And they typically go from a yellow color in fresh water to silver as they move into salt water, as their skin becomes impermeable to water and salt loss. Okay, terrestrial organisms kind of have the same problem that saltwater organisms do. The terrestrial environment tends to be very hyperosmotic. So our tissues tend to be hypotonic, right? And the air tends to pull water out of our body. So again, we're going to have to worry about water loss, right? Water being lost over our skin. So think about your skin. It's very impermeable to water loss, right? You put a drop of water on the surface and it rolls off. We do lose water through sweat as mammals, don't we? So we sweat a lot, which helps cool our bodies. Marine mammals also have to deal with water loss and salt gain, right? And so they have really, really efficient kidneys, and they can't drink salt water. Mammals cannot drink salt water. If you are stranded on the ocean, do not drink salt water. You have a better chance of survival if you drink your own urine. All right, salt water will kill you. It will kill you quickly because it'll throw your central nervous system off. We do not have the ability to remove salt, the amount of salt that's in salt water. Marine mammals can't do this either. Whales, dolphins, killer whales, they cannot remove salt from the salt water, so they don't drink salt water. Whales and other cetaceans, they can make all of the fresh water they need through metabolism. It's called metabolic water. They burn blubber or fat to make water. They have the ability to do that. We as humans can't do that, but that's an adaptation to being a marine mammal. They have the ability to make all the fresh water they need through metabolism, or they get the fresh water they need through what they eat out there in the environment. So that's how they do it. Marine crocodiles, turtles, lizards, and birds all have salt glands, like the rectal gland that I talked about with the crocodilians. They have the ability to drink salt water and then remove the salt and produce fresh water. It's a great adaptation. We, our mammals in general, do not have salt glands. We do not have the ability to drink salt water. And so don't drink salt water. Our kidneys are not efficient enough to remove the salt. We have to have a freshwater source. The main avenue of water acquisition for most terrestrial organisms is through their food and drink. Amphibians actually absorb a lot of their water through their belly skin, to the skin itself. So we'll talk about that when we get to amphibians. But that's kind of a comparison of conformers, regulators, freshwater, saltwater, and the terrestrial environments. I think that's where we'll end today. I think it's been at least 30 minutes. And we will start up with this slide here, talking about how we deal with water loss over our respiratory membranes. So have a wonderful day.